Imagine a microscopic bunker inside a tumor. It's constantly recycling its own internal waste just to survive a relentless siege.
Right, the body is actively trying to starve it out. Therapies are trying to poison it.
Exactly. But the cancer simply digests its own damaged parts to, well, generate the energy it needs to keep expanding.
It's a wildly effective survival mechanism.
It really is. What if you could find a way to break that internal recycling plant? Like, what if you could flip a single molecular switch and just starve the cancer from the inside out?
That would change everything.
Well, welcome to the OriGene Technologies podcast. In today's deep dive, for you listening at home or in the lab, we have a highly specific mission.
Yeah, they do.
We are summarizing a truly fascinating 2025 research publication from the journal Advanced Science. It's authored by Jiang and an absolutely brilliant team of researchers.
It completely reframes how we think about breast cancer progression.
It does. Yeah. And just as importantly, our mission today is to look under the hood at exactly how this science was executed. We want to explicitly highlight the OriGene Technologies products that were used to make this breakthrough study possible.
And I have to say, OriGene Technologies is incredibly excited to be a part of this groundbreaking breast cancer research.
Absolutely.
Supporting life science professionals in uncovering new therapeutic targets, I mean, that is the core of their mission. And this paper is a masterful example of how utilizing the right molecular tools reveals things that were completely hidden from view.
So true. So the mechanism at the center of this research involves a cellular process called autophagy. For anyone listening who follows oncology, you know that autophagy is basically the cell's internal recycling system.
Right.
And it's long been described as a double-edged sword in cancer.
Yeah, it's one of the most complex balancing acts in human biology, honestly, because, in healthy tissue, this recycling system is your first line of defense.
Right, it cleans things up.
Exactly. It cleans up misfolded proteins, digests damaged organelles like old mitochondria. It actually prevents early tumors from forming by keeping the cellular environment pristine.
But then the tumor gets established.
Right. Once a tumor bypasses those early defenses, it flips the script. It hijacks that very same recycling plant to survive nutrient starvation and adapt to low oxygen environments.
It basically uses autophagy to sustain its growth, even when we throw chemotherapy drugs at it. So the tumor turns a cellular janitor into a wartime supply chain.
That's a great way to put it.
And that brings us to the core discovery of the Jiang team. They found a hidden molecular break that can shut that hijacked supply chain down.
A protein called TRM38.
Right, TRM38.
Uh-huh.
Okay, let's unpack this, because this protein is changing everything we thought we knew about how breast cancer spreads. But before we get into the microscopic gears of how this break works, we need to understand the clinical reality.
We have to start with the patients.
Exactly. What happens to a patient when this molecular break goes missing?
Well, the research team began by looking at the big picture. They analyzed comprehensive patient databases and physically examined 157 clinical tissue samples from breast cancer patients.
Wow, 157. That's a really solid cohort.
It is. And they utilized tissue microarrays so they could directly compare the breast cancer tissue with the adjacent healthy tissue from the exact same patient.
Oh, that's smart. Perfect control group.
Right. And they were specifically quantifying the presence of TRIM38, which is an enzyme known as an E3 ubiquitin ligase. And the data was just glaring.
How so?
TRIM38 is drastically downregulated in the breast cancer tissues. I mean, the tumors had somehow managed to practically turn off the production of this protein.
See, if you are familiar with cellular biology, that finding alone is a major curveball.
Right.
I mean, normally when I read about TRIM38, it is strictly in the context of the immune system.
Right, exactly.
We usually think of it as a sentinel protein, something that regulates inflammation or fights off viral infections by managing interferon production. We don't usually cast it as a major player in oncology.
What's fascinating here is that historical context is exactly what makes this a genuine paradigm shift. TRIM38 is famous for its role in innate immunity. Yet, the clinical data here reveals it is also a remarkably powerful tumor suppressor.
The numbers back that up, right?
They absolutely do. When researchers stratified those 157 patients based on their TRIM38 levels, a stark pattern emerged. Patients with abnormally low levels of TRIM38 had a significantly poorer prognosis.
Shorter survival times.
Much shorter overall survival times, a faster recurrence of the disease post-treatment, and physically larger, more aggressive tumors.
Okay, I want to pause and dig into that aggression for a second.
Right.
We throw around terms like migration and invasion, But on a biological level, why does a lack of this specific protein allow a tumor to run wild?
It comes down to energy.
Right. Like, how does breaking the recycling plant prevent a tumor from physically invading other tissues?
Well, to physically metastasize, to detach from the primary tumor, reorganize its whole cytoskeleton, and literally crawl through the extracellular matrix, a cancer cell needs an immense supply of energy.
Astronomical amounts of ATP.
Exactly, ATP and fresh amino acids. And where does it get them if the local blood supply is poor?
Autophagy.
Yes, it breaks down its own internal structures to fuel that migration.
Mm-hmm.
But when TRIM38 is present, it acts as a brake on that recycling process. The researchers proved this in vitro.
Oh, they did?
Yeah. They took highly aggressive breast cancer cell lines and forcefully overexpressed TRIM38. They basically jammed the brake back into the system, and the cancer cells practically froze.
Wow.
Without the energy supplied by autophagy, their proliferation rates plummeted. Their ability to migrate and invade was severely crippled.
So the bunker is completely cut off from its internal supply chain, which leads to the ultimate question, how? I mean, how is an immune system protein wandering over and shutting down the tumor's recycling center?
We have to zoom way down to the molecular level for that.
Right. Looking at how TRIM38 physically interacts with the autophagy machinery.
So this brings us to the concept of autophagic flux. Think of autophagy as an intricate, highly regulated assembly line. There are two primary proteins you need to track.
Okay, what's the first one?
The first is SQSTM1, which is heavily cited in literature as P62. You can visualize SQSTM1 as a dedicated cargo receptor, or basically a cellular garbage truck.
The garbage truck. Got it.
Its entire purpose is to roam the cytoplasm, identify ubiquitinated cellular trash, and gather it up.
But a garbage truck is totally useless if it doesn't have a dump to drop off its load, right?
Exactly.
Yeah.
And that brings us to the second key player, a protein called LC3. LC3 is physically embedded in the membrane of the autophagosome, which is the actual recycling facility, the dump.
Okay.
So for the waste to be successfully deposited, the garbage truck SQSTM1 must physically dock with LC3, and it does this using a very specific molecular latch called the LIR domain.
LIR, which stands for LC3 interacting region. Okay, I am following the assembly line here. SQSTM1 binds to LC3 via the LIR domain, and the trash gets recycled.
That's the normal process, yes.
But wait, hold on. We established earlier that TRIM38 is a ubiquitin ligase. I mean, I have always learned that ubiquitin is basically the classic kiss of death tag inside a cell.
It often is, yeah.
Like when a ligase slaps a ubiquitin chain onto a protein, it's signaling the cell's proteasome to completely shred it. If autophagy is a recycling truck, isn't ubiquitin usually the tag that says, throw this entire truck in the incinerator?
That is a very common misconception.
Why doesn't TRIM38 just destroy SQSTM1 entirely?
That is the exact paradox that makes this paper so elegant. You are thinking of a very specific type of ubiquitin linkage called a K48 linked chain.
Okay.
K48 is indeed the universal kiss of death. But TRIM38 does something fundamentally different here. Instead of K48, TRIM38 attaches a non-degradative K63 linked polyubiquitin chain.
Oh. So it's attaching a signaling chain, not a destruction tag.
Exactly.
So the garbage truck survives.
It survives, but it is totally incapacitated. TRIM38 attaches this massive, bulky K63 chain exclusively to one tiny, highly specific location on the garbage truck, the K420 residue.
Oh, I see.
Because this bulky chain is attached right next to the docking port, it creates what biochemists call a steric hindrance.
Let me jump in there to clarify for anyone listening who hasn't taken an organic chemistry or a structural biology class recently. Steric hindrance essentially means two physical objects cannot occupy the exact same three-dimensional space, right?
That's right.
The ubiquitin chain is physically just too bulky, so the parts simply can't fit together anymore.
That is the perfect way to visualize it. It is like welding a massive, unbreakable metal padlock directly over the dumping mechanism of the recycling bin.
A microscopic padlock.
Yep. Because of the sheer physical bulk of that K63 padlock sitting on the K420 spot, the LIR domain is completely obstructed. SQSTM1 can still drive around and collect trash, but when it arrives at the LC3 dump, it physically cannot dock. The interaction is blocked.
And if it can't dock, the entire recycling system backs up. Autophagic flux is completely choked off. So the tumor cells, which were heavily relying on that continuous flow of recycled material, suddenly starve.
Exactly. The molecular brake is engaged. By simply blocking that one highly specific protein interaction, TRIM38 halts the progression of the tumor.
Man, this is incredible biology.
Yeah.
But, if you are a researcher listening to this deep dive, you know that discovering a microscopic steric hindrance, a molecular padlock on a single amino acid, that doesn't happen by just staring through a standard microscope.
Definitely not. Proteins are largely invisible, and their interactions are so transient.
Right. To map this exact protein interaction, the researchers had to literally hijack the cancer cell's internal machinery. They had to manipulate the genetics to see what was happening.
Which brings us directly to the methodology.
Exactly, and the tools they used to achieve this.
The methodology is an absolute masterclass in molecular biology. I mean, you simply cannot peer into a cell and visibly see a K63-linked ubiquitin chain sitting on a K420 residue.
You wish you could, but no.
Right. You have to isolate these proteins, extract them from the chaotic soup of the cytoplasm, and rigorously test their interactions biochemically.
And this is where we have to highlight the specific OriGene Technologies product that acted as the absolute linchpin for this entire phase of the study.
It was crucial.
It's the PCMV6 entry mammalian expression vector. And for anyone taking notes for their own lab right now, the catalog number is PS100001. Honestly, framing this vector as a mere product doesn't really do it justice.
It really doesn't.
It was the molecular solution to an enormous visibility problem.
Let's break down the lab work for the listeners. To unequivocally prove that TRIM38 binds directly to SQS-TM1, the team needed to massively overexpress TRM38 in mammalian cells.
So they needed a reliable vector.
Exactly. They utilized HEK293T cells, which, are human embryonic kidney cells known as exceptional factories for producing proteins, as well as actual breast cancer cell lines.
Okay.
They took the human TRM38 gene and cloned it directly into the OriGene PCMV6 entry vector.
Here's where it gets really interesting. The design of this vector is brilliant, because if you were trying to pull a specific protein out of a cell, you need an antibody that recognizes it, right?
Yes, and native antibodies are notoriously finicky. They often lack the specificity required for clean data.
Right, but the brilliant feature of the PCMV6 entry vector is that it automatically appends a C-terminal MiCDDK tag to whatever protein you are expressing. And just to clarify terminology across different labs, that DDK tag is the precise molecular equivalent of the highly popular flag tag.
That specific tag changes the entire game for an experiment like this.
It really does.
Because the OriGene vector reliably attached that flag tag to the end of TRIM38, the researchers suddenly had a highly specific, universally recognized handle to grab onto.
No more guessing with native antibodies.
Right. They didn't have to hope their antibody worked. They could use commercially available, highly potent anti-flag antibodies to clearly detect TRIM38 in their Western blots.
It's essentially putting a bright fluorescent neon vest on TRIM38 so you can spot it immediately in a totally crowded room.
And beyond just spotting it, they use that neon vest for co-immunoprecipitation or co-IP assays.
Oh, to see what it interacts with.
Exactly. They basically used those anti-flag antibodies attached to microscopic agarose beads, like a set of highly specific tweezers. They reached into the lemmel's cellular extract and pulled the tagged TRIM38 right down to the bottom of the test tube.
And whatever is bound to it comes along for the ride.
Yep. Once they isolated TRIM38, they washed away the rest of the cellular material and checked to see what was stuck to it. Sure enough, SQS-TM1 was firmly attached.
They also used this vector to play a game of, molecular Jenga, didn't they?
They did, yeah.
Because they didn't just express the full protein. They created truncated, mutated versions of TRIM38, deliberately chopping off different structural domains to see at what point it would stop binding to SQS-TM1.
And through the robust expression provided by the OriGene vector, they systematically tested these tagged mutants. They successfully mapped out that the CC and press-spry domains of TRIM38 are absolutely essential for physically interacting with SQS-TM1. Without the reliable, high-yield tagging of that specific vector, proving these physical domains would have been vastly more difficult and, honestly, prone to so much background noise.
Okay, so the researchers used the OriGene vector to prove the binding mechanism. They successfully located the padlock. But, if you are reviewing this data with a critical eye, the next logical question is, how do we know for sure that targeting this exact K420 spot is the thing actually stopping the tumor?
That's the vital next step.
How do we prove it is the actual choke point and not just a coincidental interaction.
To prove causation in molecular biology, you rely on rescue experiments. You intentionally break the system you just discovered to see what happens, and then you try to rescue the original function to prove you truly understand the mechanics.
Show your work, essentially.
Precisely. So the researchers went back to the SQSTM1 protein and specifically mutated that one critical spot. They changed the lysine amino acid at position 420 to an arginine.
Ah, creating a mutant known as K420R.
Right. Because arginine has a similar positive charge to lysine, the protein doesn't completely misfold and fall apart. However, arginine cannot accept a ubiquitin chain.
Oh, wow. So with that tiny chemical alteration, TRIM38 had literally no place to attach its padlock.
Exactly. The break line was cleanly cut, and the biochemical results were immediate and undeniable.
What happened?
When they introduced this K420R mutant into the cells, autophagic flux violently restarted. The recycling plant reopened.
And the physiological result for the tumor cells.
They immediately began proliferating, migrating, and invading all over again. The powerful tumor suppressive effect of TRM38 was completely bypassed just by changing that one single amino acid.
That is why.
It proved unequivocally that the K420 steric hindrance is the vital choke point.
Okay, that level of microscopic proof is incredibly satisfying. But if you're a patient or a doctor, so what does this all mean? How does finding a microscopic choke point translate to eventually saving lives?
If we connect this to the bigger picture, it opens up entirely new, highly targeted avenues for combination therapies.
Combination therapies. Okay.
The researchers took this hypothesis into in vivo mouse models. They implanted tumors in mice and forcefully overexpressed TRM38 to engage the brakes. But then they added a pharmacological element, hydroxychloroquine, commonly known as HCQ.
Wait, HCQ is an existing widely known drug.
Yeah.
It's historically used for malaria and various autoimmune conditions, right?
Yes, it is. And it is currently undergoing intense clinical trials as a potential autophagy inhibitor for cancer therapy.
Oh, I didn't realize that.
Yeah, HCQ also attempts to shut down the cellular recycling plant, but it does so through a completely different mechanism. It raises the pH inside the lysosomes, essentially neutralizing the acidic environment so the digestive enzymes inside the dump just can't function.
So they hit the tumor supply chain from two entirely different angles simultaneously.
Exactly, and the synergistic effect was incredibly potent. Tumors in mice that received both high TRM38 expression and HCQ treatment were dramatically suppressed compared to either treatment alone.
That's a huge result.
It is. The TRIM38 overexpression heavily sensitized the tumors to the baseline anti-tumor effects of the HCQ.
But I have to push back here just a little. Curing cancer in a controlled mouse model is a notoriously long way from treating a human patient. Are there systemic risks to this approach?
Oh, absolutely.
Like if we eventually give a human a systemic drug that shuts down autophagy, or a booster that artificially maxes out TRM38, won't that choke off the recycling plants in healthy cells too? What about neurons or heart muscle cells that desperately rely on autophagy to clear out toxic plaques?
That is the most critical hurdle in translating this to the clinic, without a doubt. It's all about finding the therapeutic window. You have to inhibit autophagy enough to starve the highly demanding tumor, but leave enough baseline function so healthy tissues don't undergo toxicity.
Right.
This is why targeted delivery is the next frontier. The paper suggests two major future therapeutic strategies. The first is screening for small molecule drugs that act as specific TRM38 agonists.
Basically a drug that selectively turns the gene on within the tumor microenvironment.
Exactly. Flooding the cancer cells with molecular breaks.
And the second strategy?
The second is utilizing PROTACs, which is truly cutting-edge pharmacology.
PROTACs, proteolysis targeting chimeras.
Right. PROTACs are engineered bifunctional molecules. You are essentially building a molecular matchmaker.
How does that work here?
You could theoretically design a PROTAC with one end that specifically binds to TRIM38, a chemical linker in the middle, and another end that selectively binds to SQSTM1.
Oh, I see.
By utilizing induced proximity, the PROTAC forces the two proteins together, artificially applying the K63 padlock to choke the tumor, bypassing the cell's natural regulatory signals entirely.
Honestly, that sounds like science fiction, but it is exactly the reality of modern drug design. And none of it is possible without first mapping the basic molecular pathways. Which brings us full circle on today's deep dive.
It really does.
We have seen how TRIM38 operates far beyond its traditional role as an immune regulator. It is a vital structural tumor suppressor. It literally starves breast cancer cells by dropping a K63-linked padlock onto their nutrient recycling trucks.
And it is essential to reiterate that mapping this incredibly nuanced, life-saving pathway was fundamentally enabled by the robust expression and tagging capabilities of OriGene's PCMV6 entry vector,
Absolutely.
providing researchers with the precise, reliable molecular tools necessary to pull these invisible protein complexes out of the dark and visualize them on a Western blot. I mean, that's the critical first step in moving from basic science to clinical therapeutics. And OriGene Technologies is immensely proud to support the life science community in this exact endeavor.
It perfectly highlights the vital partnership between the brilliant scientific minds designing these experiments and the engineers building the tools to execute them.
It absolutely does. However, as we wrap up this analysis, this research leaves us with a profound, lingering mystery. Something the authors themselves noted in their discussion, which really highlights the sheer depth of what we still have to learn.
Okay, what is the lingering mystery?
This raises an important question. The researchers noted that there is another enzyme inside our cells called TRIA2. Interestingly, TRIAF2 also adds a K63-linked ubiquitin chain to the exact same K420 spot on SQSTM1.
Wait, I want to make sure I understand this. The exact same chemical tag, linked in the exact same way, attached to the exact same amino acid.
Yes. But here is the massive catch. When TRIAF2 applies that tag, it causes a completely different cellular outcome.
Different how?
Instead of acting as a steric hindrance that blocks the protein, it somehow leads to the degradation and destruction of the protein.
How is that physically possible? If you are applying the exact same chemical padlock to the exact same latch, shouldn't the mechanical result be identical?
You'd certainly think so. But it points to a concept biologists call the ubiquitin code. It suggests that the language the cell uses to signal what to do with proteins is breathtakingly complex. If the same tag at the same location triggers entirely opposite behaviors, it implies that the outcome is dictated by invisible, highly dynamic 3D structural folds.
Wow.
Even with incredible AI tools like AlphaFold, which are excellent at predicting static protein structures, we struggle to predict the dynamic, real-time flexibility of these ubiquitin chains. Does the chain fold slightly to the left? Does it interact with a hidden pocket on a nearby protein? We are only just beginning to comprehend the sheer four-dimensional complexity of these microscopic machines.
That is wild. It's the biological equivalent of finding out that a word in a sentence can mean two completely opposite things entirely based on, like, the font it happens to be printed in.
Exactly.
It just proves that for every mechanical door we manage to unlock with incredible tools like OriGene's expression vectors, there is a whole new echoing hallway of mysteries waiting behind
There
it.
really is always more to explore.
Well, if you are listening to this, you have successfully navigated another dense, incredibly complex biological landscape with us. Thank you so much for joining us on this deep dive into the microscopic battles happening inside ourselves.
Thanks for listening.
And a massive thank you to OriGene Technologies for supporting the level of scientific discovery that makes these conversations possible. Keep asking the hard questions, keep looking closer at the data, and we will catch you on the next deep dive.